Hello, my name is Anne Kuipers and I'm from Hasselt University, and I'm glad to introduce you to the lecture series on the redox balance. And in this lecture I will focus on the redox balance as a central mechanism in plant stress responses. As you can see on this slide, plant research is conducted at different biological organization levels, ranging from smallest molecule until field experiments and field trials. But why is it now so important to study plants? Plants have many functions and we cannot survive them. And as you can see, plants are the primary producers in which they inhale carbon dioxide and take up water from the soil, and they use the energy of the sun through photosynthesis to produce oxygen and carbohydrates like sugar. So this is very important for us. But besides this, plants can also serve as a natural resource for food, vegetables and fruits, feed for our cattle, fibers to produce, for instance, textiles, and fuel to have our biofuel. And to take this all into account, plants have a crucial role within the ecosystem services. You can see 4 parts, which is provisioning, regulating, supporting, and also cultural. And if we look to the provisioning services, it's clear that plants are essential to provide us these resources, as mentioned before, as food, as raw material, but also to make medicines, for instance. So many pharmaceuticals and traditional medicines are derived from plant compounds, and of course this fuel. They are important in regulating, so maintaining the ecosystem balance, because they regulate essential ecological processes that sustain the environment. For instance, carbon sequestrations. As we mentioned in the first place, plants perform photosynthesis, and in this way they absorb carbon dioxide and reduce this greenhouse gas and help in mitigating climate change. They also regulate air quality, because plants can filter pollutants and improve air quality by absorbing gases and trapping particulates. Also for the water cycle regulation, it's important because they help in reducing runoff of water, stabilizing watersheds, and also then, of course, the soil health itself, because plants prevent soil erosion and as such improve soil fertility and support microorganisms, which are critical for nutrient cycling. So in general, they regulate also the climate by controlling the temperature, humidity, rainfall patterns. If we then look to the left side, plants have also supporting services, because of course, they have the primary production capacity, but they provide also habitat for a lot of organisms, like for instance soil microbes, but also some bigger animals. And as mentioned before, the nutrient cycling is a very important supportive service. And that what might be a little bit less known, but plants are very important in cultural services. They have an aesthetic value, so they give landscape beauty, a natural beauty, which can inspire artists, for instance. But of course, they have a very important recreational opportunity. And some of the plants that we know have also some cultural significance, like symbolic flowers, for instance, that are really involved in many cultural traditions. As human activities, we put these plants also at risks. If we think about climate change and environmental pollution, for instance, this poses a severe threat to plant health. And if we look at the climate change, in the first instance, then we know we have extreme weather events. The global temperature is increasing, so we get more extreme weather events like heat waves, but also precipitation extremes. The sea level is rising, and of course we change land use for our plants. And what I show here is actually, on the left side, it's a prediction of the Intergovernmental Panel of Climate Change, in which they put forward different scenarios in which if the temperature rises within this scenario, what can be the problem by the end of this century. And then on the right hand side you can see what is the impact in between these different scenarios related to, for instance, maize yield production. So you can see on the globe chart that the more we go to the more extreme events, the worse it gets for maize yield production, because we see more pink appearing on the map. So it's clear that we have to do this research to make sure that we can mitigate these problems and still have good plant growth. What is also important about plants is that they are sessile organisms, so they cannot run from stress conditions, and so they have to have a lot of details, a lot of characteristics that can help them with coping with this environmental stress. And we needed to develop strategies to mitigate environmental problems, but also to maintain and support biodiversity. As mentioned before, plants are a great habitat for lots of other organisms, but also we want to maintain biodiversity. We want to improve plant stress resilience so that they are capable of coping with environmental stresses. We want to improve crop yield, but also the nutritional value is important, and a big example is golden rice production, for instance. And then, as I mentioned before, we want to remediate polluted soils, for instance, through phytoremediation, but also make agriculture and industry more sustainable. And then if we have these natural components, plant components, we can use them for the production of, for instance, biofuels, but also medicines. But how do we start to study plant stress? Therefore, we use the scientific method. So we ask a question, what are we wondering? And then we do some background search. From this background search, we construct a hypothesis that we will test with an experiment. If everything works nicely, we can further go down, analyze the data and draw conclusions. And from these conclusions, we can say, okay, the results align with our hypothesis, and we communicate the results, or they do not, or not completely align with the hypothesis. And our experimental data will become the background for future experiments. If our procedure, our experiment wasn't working, we have to do some troubleshooting for the procedure and make sure that the experiment is conducted clear. So in the case of plant stress, we ask a question about what is the impact of abiotic stress factors on our plant growth. And for this, for the abiotic stress factors, I also want to refer to the module on new emerging pollutants. So we are wondering, if plants are exposed to abiotic stress factors, what will be the consequence? And therefore, we look first at the growth defense trade-off. So it means that if plants have to invest in defense because they have to cope with environmental stress, they use the energy for defense and cannot use this energy for growth. So this lecture will focus on this growth defense trade-off that plants face when they are exposed to abiotic stress factors. But as I mentioned, it's important that your experimental design is very compelling and also addressing the hypothesis you put forward. And for the experimental design, we can go from highly controlled to environmentally realistic conditions. And here you see an overview of different aspects on how we can conduct environmental research from strictly controlled, and that's most often in vitro and in climate chamber researched, to more realistic, going to soil experiments, greenhouse facilities, for instance also the ecotron, which is very climate change controlled condition but realistic to the environment, to really open field experiments. Then also, if you want to go from highly controlled to more environmentally realistic conditions, we can go from single stresses to multifactorial stress conditions, because a plant is hardly ever exposed to only one stress condition. So therefore, it's important in future research to take multiple stresses into account. And of course, it's very interesting to work with model organisms because they are often easy to work with, easy to handle. We have a lot of information from the molecular side on these organisms, and often they have a short half-life, a short life cycle. But from there, we can go to economically interesting crops. So the experimental design really depends on your research question. And the research question can then range from really fundamental research to explore new elements, over translational to applied research, in which you really get closer to real life systems. And aspects that we have to consider are: which organ do we need to answer our question? So which organ do we have to sample? Will it be roots? Will it be the leaves, whole seedlings, whatever? How will we expose the plants? At which developmental stage? Are we really interested in the young, so seeds and young seedlings, or more into the mature plant? How long will we expose the plants, from acute, short-term, to chronic, long-term exposure? And how many replicates do we have to take to get really a significant outcome, significant results? And for this, we have to take into account the expected effect size and the variability, as well as costs, time, and space that we have. We can also see at which biological organization level we want to focus. Is it the cells, or are we going over the organs to the entire organism? That's important to envision. And so all your experimental design really depends on which biological organization level you are focusing on, which abiotic stress factors, combinations. This will say, okay, that's the best experimental design to use to study this growth defense trade-off. And if we are looking into these different levels, we can see that the redox balance is a very important central mechanism in this system. And to get better acquainted with the redox balance, I refer to the lecture on the introduction to oxidative stress. So if we see the redox balance here, which is a balance between antioxidants on one hand side and reactive oxygen species on the other hand side, it can lead to two characteristic outcomes. It is the oxidative damage on one hand side, which is losing the balance between free radicals and antioxidants, and oxidative signaling, which is using this balance between free radicals and antioxidants to steer processes. And this is also the case in plant research. But how do we investigate now this redox balance? We can do it directly by investigating biomarkers of the redox balance, and it's really directly analyzing the free radicals or reactive species, and on the other hand, antioxidants, which can be enzymes or metabolites. And for these biomarkers, I refer to the advanced class on the analysis of reactive oxygen species and antioxidants. On the other hand, we can also indirectly investigate the outcome of this redox balance, and there we have a strong interplay with other mechanisms. And in our case, we look to this growth defense trade-off in plants. The growth defense trade-off can be investigated at different levels or using different approaches: a holistic approach, studying all stress responses at a certain level as a whole, or we can use targeted approach. Looking at this holistic approach, we have the omics technologies at different biological organization levels, the genes, genomics, the mRNA or the expression at transcriptomic level, and here I also refer to the gene expression analysis course that is provided in this module. And then of course we have also the proteins, metabolites and phenotypes, and you always go and look at a different level, biological organization level in the plant. If first we have the oxidative outcome and we look into the damage part, then the analysis of cellular damage is very important. And also in this module on technologies, I want to refer to the COMET essay, which was provided in this module. So we can analyze DNA damage, protein oxidation, lipid peroxidation and membrane leakage, and finally cell death. For the growth of cells, the analysis of cellular growth, we can look at the cell division and endoreplication. And cell division and endoreplication can be monitored using flow cytometry, which is also provided in an advanced class in this lecture module. If we then start looking from the most total biological organization level, then we look at the total organism and we can do phenotypic analysis of plant growth. For this, we can look at root growth using vertical agar plates, for instance, but we can also follow vegetative growth and reproductive growth by looking at the upper soil parameters, like for instance the rosette diameter and area, the number of leaves, and for the reproductive growth we can look at, for instance, inflorescence, emergence and length, the silique number, seed counts, and so on. But of course, this is all very labor intensive because you have to do a manual monitoring, and the temporal resolution is of course then limited because you can only do it a limited amount of times during your work. For that, we have also automated monitoring via multispectral imaging, and in this case we use a multispectral camera which can really monitor different parameters at once. It has the benefit that it has a high temporal resolution. You can do it multiple times per day, and you can also add other parameters related to plant health, like for instance pigments, photosynthetic efficiency, and the NDVI, which is related also to plant health. If we then dive into the physiological analysis and we look, for instance, in this regard to the leaves, we can study the relative water content, stomatal conductance, chlorophyll content, and photosynthetic efficiency at leaf level. And then, as mentioned, biochemical, we want to look at more the metabolites. Then in the stress responses of plants, phytohormones play a crucial role in steering the response at organism level of these plants. But also secondary metabolites are very important because these secondary metabolites help the plant to overcome stress, and you can see sulfur-containing compounds, nitrogen-containing compounds, phenolic compounds and terpenoids, but these are, as mentioned and indicated, important in plant defense, but they are also very important as a resource for human health because they have anti-inflammatory, antibiotic and antioxidant properties, as well as anti-cancer, painkiller and detoxification characteristics. And then if we want to dive even further into the molecular analysis, then it's really important to have crops or model plants for which genetic resources are available, and the most important or most known plant in this regard is Arabidopsis. We have plenty of mutants and transformants available of these plant as well as natural accessions. Furthermore, we have lots of molecular tools available which we can use in these model plants so that we can really dedicate the experimental design to what we want to investigate. And in this regard, also CRISPR and CRISPR-Cas has gained a lot of interest over the last years. So it's really important that you have a very good experimental design so that you really decipher which experimental setup you will use, what is the stress intensity that you will use, because this, through perception of the stress, signaling responses and an altered metabolism, will define whether you have damage or acclimation of your plant, which is referred to as the stress resilience of your plant to these abiotic stress factors.